Hello world, and thanks for joining me here in this corner of the internet. My name is Owen Chapman. I'm a third-year graduate student at the UCSD Bioinformatics and Systems Biology PhD program. And I've adapted my poster to presentation format so I can provide some verbal context to each component. I'll begin by motivating my work. As is the case with many cancers, there are many possible mutations which can give rise to the malignant pediatric tumor of the cerebellum known as Medulloblastoma, or MB. On the right are summarized some of the features associated with the four main molecular subgroups: Wnt, SHH, group 3, and group 4. On the left, I show the distribution of MB cases affected by mutation of specific genes. A key feature you'll notice here is that while a few key driver genes may be able to explain some plurality of cancers, there is also this long tail of low-frequency driver mutations, which individually account for only a small percentage of cases each, but collectively account for the majority of cases. One of my ongoing projects has pursued the broad hypothesis that while individual genetic mutations may be distinct, collectively, this long tail may be targeting one or a few oncogenic cellular pathways. To explore this hypothesis, I've been combining genomic, transcriptional, and epigenomic data from patient genomes to build models of transcriptional regulatory circuitry in medulloblastoma. To gain a clear understanding of what is going on in MB, we've assembled a cohort of primary MB samples and characterized them using many different sequencing modalities. Here, I will focus principally on ATAC-seq and RNA-seq. ATAC-seq measures chromatin accessibility across the genome. Generally, chromatin is accessible when it is transcriptionally active as a promoter, enhancer, repressor, or insulator. We use ATAC-seq to catalog transcriptional regulatory regions in each sample. Very briefly, we performed NMF consensus clustering on signal strength for 56,000 accessible regions in our ATAC-seq cohort. At the optimal cluster number of k equals 7, we see that the accessible landscape of these samples reflects their MB subtype identities, indicated by the color bar on the left: SHH, G3, G4. There were no Wnt samples in our cohort. Subclusters appear to correlate with ATAC-seq experimental quality. To associate regulatory regions with their target genes, we perform a correlation analysis between gene expression and nearby chromatin region accessibility. An example is shown here. We have a distal region upstream of the MYC locus, which is active only in some MB subtypes. Correlation to gene expression of nearby genes shows a strong association with MYC expression. To limit the number of hypotheses tested, we examine only correlations which occur within the same TAD, or topologically associated domain. Next, we identify the transcription factors which are active in each MB subtype. To accomplish this, we have adapted the popular gene set enrichment analysis, or GSEA, to epigenomic data inputs. Here's how it works. On the left, in Figure A, we take all regions identified by ATAC-seq and rank them by differential accessibility in a given subtype versus the others. Also in Figure A, we annotate each accessible site if it contains a binding site for a specific transcription factor, or TF for short. Now, if there were no association between subtype and TF, we would expect a random distribution of binding sites across this ranked list. In this toy example, however, you can see clearly that the TF binding sites are enriched in the most differential regulatory regions. We quantify this enrichment using a running KS statistic shown in Figure B. In Figure C, we can estimate significance of this statistic by permuting the binding site annotations across the accessible sites and recalculating this enrichment statistic for each. We now have all the components to build a network model of transcription regulation in each medulloblastoma subtype. 1, a knowledge of which transcription factors are active, and 2, a set of strong correlations between TF binding sites and nearby genes. From this, we assemble network models of the core transcriptional machinery active in each MB subgroup. The core network for the SHHA subtype is consistent with known regulatory biology in SHH medulloblastomas, highlighting, among others, neural differentiation factors, TCF3 and TCF12, as possible upstream regulators of well-established SHH subgroup oncogenes, GLI2 and ZEB1. The core network for G3A subtype re-identifies the known group 3 medulloblastoma core regulatory factors with remarkable fidelity, including OTX2, HLX, LHX2, MYC, and others. Among the best supported regulatory relationships is the OTX2 to MYC regulatory axis, which is well described in this subgroup. We also see possible upstream regulators of these oncogenes, including a self-regulatory feedback loop for OTX2 and a possible role for NeuroD1 in determining cell fate in this subgroup. You may have noticed that I didn't talk at all about our Hi-C data. Hi-C detects genome-wide interactions between any two genomic loci. Our next steps will use Hi-C data to resolve chromosomal rearrangements and to further elucidate enhancer-promoter interactions. Thanks for listening, and I'd also like to thank the good American taxpayer for supporting this work. Joe Mezarov and Lucas Chavez are my PhD advisors, and you can expect more exciting work from us in the near future.